This is a recording of Chapter 11, Synapsids and Sauropsids. Hopefully you remember so far from class that the synapsids are the mammal groups and that the only extant members of these groups are mammals. The sauropsids, however, contain the turtles. These scaly reptiles, our tuataras, are lizards and snakes, crocodilians, including the crocodiles, the alligators, caimans, the birds, and our extinct dinosaurs. As our animal groups move from amphibians to terrestrial in their lifestyle, the terrestrial environment did provide new ways for living, but it also had some requirements with it. Eggs were now able to work for reproduction, but the egg needed to provide that amniotic, that aquatic habitat for the development of the fetus. But eggs allow for larger offspring, at least the eggs that can be laid on the terrestrial habitats. Locomotion is also going to be very different. We'll see that in order to sustain locomotion that is possible on terrestrial surfaces, you'll also need to increase the amount of feeding that is done. And with locomotion and feeding, we'll discover that more advances arise, including changes in respiratory structures and a move towards endothermy and away from ectothermy. We'll also see that there are social interactions as we go through the different groups of the synapsids and the sauropsids that are very different from social interactions we've seen in other groups. This is really going to include social behaviors and parental care advances that we haven't seen so far. The sauropsids and the synapsids appear to have evolved their lines separately rather quickly. And because of this quick split, there's little overlap in the lifestyle adaptations for their lifestyles. Both have very different types of ways of approaching things. And they represent remarkably different and independent evolution pathways of terrestrial life forms. And because of this, they display parallel evolution. Respiratory and locomotion could be in conflict if the body wiggles a lot as it's moving, and in doing so squishes the lungs. But we'll see that our sauropsids in their evolution have gone towards bipedal with ribcage contractions for controlling respiration, whereas the synapsids have relied on the diaphragm movements. We'll see that both groups can deal with high rates of oxygen consumption differently through the development of either a through flow lung or a tidal flow lung. For endothermy and keeping warm, they'll use different types of strategies, relying on feathers or hair or fur in order to assist in the development of endothermy, but not as the full reason for the development of endothermy. And excretory systems are also different for either uric acid or urea. We'll also look very briefly at some differences in their sensory systems. Living on land gives the opportunity for sustained locomotion, different locomotion though in water, and very different from your amphibians. Running on land is going to allow for a very different style of locomotion, and it's going to demand a lot from the organisms because running is going to require the ability to move limbs rapidly and to generally move them over a long distance for a long period of time. This means that they're going to need muscle structures that allow this, a body structure that allows this. We're going to see the movement of the limbs from a sprawling stance to the limbs being underneath the body trunk. It's also going to include different types of connections between the spine and the body. The backbone is now going to need to be kept stable. That stability is going to be important if they run for more than just a short distance, because breathing is going to be impacted by running if there's not a stability to the spine. Lungs would compress as the body moves side to side, and you wouldn't be able to get good breaths in. There needs to be a steady supply of oxygen in order to support the muscles with the amount of work they'll be doing for long distance locomotion. And also another reason that we'll need the amount of oxygen is because of metabolism. You're going to need to create a lot of energy, and the creation of ATP is going to require high sustained oxygen production. We'll see that in order to have sustained locomotion on land, we're going to need to separate our respiration and our locomotion processes from each other. The way that we do this is a little different depending upon the group. At the top, we have a lizard or a sauropsid. At the bottom, we have a dog, synapsid. You can see that the lungs are compressing in the top as they move from side to side. But if you can take advantage of this to move the air through the body, then perhaps it's not a disadvantage, but in fact an advantage to use it to create the pressure that you need, the negative pressure or the positive pressure, to move air around the lungs and therefore increase oxygen consumption. The dog is going to have a different structure. Instead of moving air between lungs, you're going to be using the lung movements and the diaphragm contractions and expansions along with the movement of the whole body to increase the pressure to move air in and out of the lungs. These are two very different approaches to increasing the oxygen that can get into the body, but both work well for the organisms that use them. Our earliest synapsids were likely short-limbed with a sprawling posture and long tails. As they developed, and evolved, we have the therapsid group, which is a more derived part of the synapsids that our mammals eventually came from. They developed a more upright posture, moving the limbs underneath the trunk. So they went from looking more like the sprawling stance of a crocodilian or a lizard to having more of an upright stance, upright posture at least, of a cat, a dog, a rabbit. And this allowed them to move without bending the trunk as they developed this upright posture. The diaphragm evolved or developed within this group, and it helped to resolve that locomotion respiration issue of how do you move enough oxygen in and out of the lung. The diaphragm creates negative pressure for breathing in and positive pressure for pushing air out in exhaling. And it allows this change in pressure of the lung to drive the inhalation-exhalation processes. And it allows it to drive them in a way that's energetically efficient and muscularly efficient. Respiration and locomotion could now be synergistic. They were working together. Faster movements of running aided in moving the diaphragm faster, allowing more oxygen to move in and out, more air to move in and out of the body and increase your oxygen intake. We do not, however, see this in bipeds like humans, or perhaps not as much in kangaroos and things like that either. This synergistic relationship between respiration and locomotion is, however, common in the quadruped mammals still. And when we look at their respiratory rate, it is actually a multiple of their stride interval, which means that in order to be more efficient, they don't increase the number of strides while they're running, but they actually increase the distance of a stride while they're running, because that allows them to keep the locomotion and the respiration in sync as opposed to getting them out of phase. This is looking at the early therapsid groups, and you can see that movement towards a more upright posture as opposed to a sprawling stance. You should also take note of the differences in the spine, the differences in the ribs too. You do see a very large movement here in rib differences as the rib cage gets shorter in the distance of the spine that it covers, as well as the ribs themselves do change in how they're supporting the body. Sauropsids will do this locomotion respiration combination a little bit differently. Those that are bipedal with locomotion do not have any movement of the trunk, They're going to have lung structures that are quite different than probably what you're expecting, but they don't have a linkage between respiration and locomotion problems, similarly to how humans do not. The earliest sauropsid quadrupeds were very similar to the early synapsids, where they had this sprawling stance, and crocodiles still maintain this sprawling stance. And because of this, poor crocodiles, if they move too fast, they can actually start to hyperventilate. So crocodiles, while they can run fast, do not run fast for long distances, which means if you have to outrun a crocodile, you can probably run a longer distance than it can, but you may not be able to run faster in the shorter distance than it can. The reason that they hyperventilate if they move too fast is because they use their trunk to move air in and out. We'll see what that looks like in a moment. They do this by using the movement of the ribs, the liver, and rotation of their pubic bones. These pelvic movements and rib movements aid in lung ventilation. We do see that birds still utilize some of these movements, and since crocodiles used them, we assume that the dinosaurs likely did as well, as they would have had common ancestry. So this would be an evolutionary hypothesis here, that dinosaurs probably utilized these movements for respiration. This is looking at the crocodile respiration. On the top, we have inspiration or inhalation, and at the bottom, expiration or exhalation. As the alligator breathes in, the liver is going to actually be pulled backwards. The pelvic bones are going to rotate generally in a ventral way, and the volume of the lung is going to increase. As they exhale, the bones are going to rotate dorsally, and air is going to be forced out of the lung, and the liver is going to move along with it. So the liver is going to move up during the exhale process. So the liver here is working not the same as the diaphragm, but it is kind of aiding, and it may not really be aiding in the process, but it's acting in a similar location to the diaphragm, and we're using it to figure out how the lungs themselves are moving, the movement of this region of the body. When we look at our sauropsids and our synapsids, we see that minor technical difficulty there. For lung ventilation and locomotion relationship, it's also important that we consider that as sauropsids and synapsids continue to evolve, one of the things they started to do was being able to move farther and perhaps faster. But in order to do this, they really did have to effectively separate this respiration locomotion problem. For the synapsids, that was easily done by the diaphragm, but the sauropsids, it really did depend upon the group, and we'll look at this a little bit more in depth as we go through these sauropsid groups later. This is looking at that change in respiration. This may help you as we go further into these groups later this semester to see where the differences are between the groups and how they're able to do the respiration. But once again, it's something we'll come back to with most of our groups. In order to increase gas exchange, it wasn't just enough to separate locomotion and respiration. You actually needed to have better respiratory surfaces. And ventilation of lungs by ribs is ancestral in our ammonites. The trachea is also ancestral. And at first we see these very simple lungs, which are ancestral. But as the oxygen demand increased, we see an increase in lung complexity. Our species that require the most oxygen tend to have the most complex lung structures. The sauropsid and the synapsid groups have very different ways of doing this. both have developed lungs with elaborate air passages. And there's a chance that this development or this movement towards these more elaborate lungs and a life that demands more oxygen may be in fact related to an environmental thing. That the environment had higher levels of oxygen in the atmosphere, allowing then for more oxygen to be used. And if you could use more oxygen, well, you needed to be able to take more of it in. Once you had more oxygen, you could move further and faster, and life was better. So there is some environmental and not just genetic and physiological control over this. So here we see an interplay between the environment, genetics, anatomy, physiology, and development. So what are these methods they use to increase their gas exchange? Well, here we see the ancestral lung, and then the two different structures that we have in our groups. Your synapsids went to the alveolar lung. The sauropsids went to the faviolar lung. You can see they do look different. And this is a, for the faviolar lung, it's quite generalized here, but we will have a picture of what this might look like in a bird in a few slides to get a better idea. But first, we're going to start with the lung you're most familiar with, the alveolar lungs. These have pretty elaborate sac-like lungs with progressively smaller passages. You go from the trachea to the bronchi to the bronchioles, and then eventually ending in your alveolar sacs. They have thin walls, as you know, one cell really wide, that allow easy exchange of oxygen from the air in the lung into the blood in the body. And it moves oxygen and carbon dioxide very fast across this single type cell layer. But because of this, and because of how complex they are, you get these very small air sacs. So small, in fact, that they would basically collapse without the secretions that the lungs make, which help in reducing the surface tension and then kind of the weight and the structures of the water. And these alveolar lungs work very well. These are the lungs you're familiar with, the lungs you've likely dissected in Bio 152. The valvular lungs are a bit different. They can also be called separate lungs because they have a common space that will then develop into smaller spaces. So instead of just having two lobes, we're going to have more lobes generally of the lung. Most have a tidal flow in and out, similarly to what we have, where you inhale, fill the passages, exhale, the passage is empty. But birds have a flow through system where they have air sacs throughout the body that are divided into anterior and posterior sacs with cavities where the air will move through them. And they have air sacs where it will flow from one section to the next, to the next, to the next, as the air flows through the whole body and then back out, as opposed to just going that simple in and out of tidal flow. This does create a cross-current exchange system similar to what we talked about in the gills. The valvular lungs can just be shallow chambers in the walls, near the bones and things, or they can be more complex with pretty extensive subdivisions throughout the body. And here we have one of the more complex ones. Your birds have paired lungs. So you wouldn't just see one side of the lung here. You're only seeing one side of the lung here. And the oxygen is actually going to move, the air is going to move as they're taking oxygen out of it and putting carbon dioxide into it throughout these systems. That probably seems a little strange. But it's going to move something like this. In the first inhalation, it's going to fill some sacs. As they then exhale from that, it's going to move through another area. As they inhale again, that air is going to go into another area. And then as they exhale out, it will then go out. So while there is in and out movement perhaps in the trachea, we see that through the system, air is only flowing in really one direction. So birds have this one-way flow of air. And what it really allows is for them to minimize the dead spaces in the lungs. It allows them to reduce the stale air in there, allowing a more complete exchange of the air in their bodies, and for more oxygen to be exchanged rapidly. We believe this is what enables birds to breathe at high altitudes and get the most oxygen out of that air. Because birds fly at ridiculously high altitudes that our lungs are incapable of getting enough oxygen from without some form of an oxygen delivery system, usually an oxygen mask or something like that. But birds, they can handle it, probably because of this modification. Dinosaurs had air sacs as well. They had cavities in their body with openings of air sacs. And what we find are grooves on their vertebrae indicating where these sacs were located, similar to the grooves that we find on bird vertebrae. Those with long necks, we find these grooves from the neck all the way through to the tail area, suggesting that the longer organisms had more air sacs, and that a one-way flow of air went through the entire body. The theropods had a pneumatic vertebrae with cervical and abdominal air sacs, and they probably were more like us with a flow through ventilation than the birds with a one-direction airflow. But it's not just enough to have oxygen. You also have to be able to move that oxygen around. And better locomotion not only requires more oxygen, but more oxygen delivery then to the muscles, so that the muscles can do more metabolism to create more ATP so that you can move further distances. So you need rapid transport of oxygen then around the body. This is going to require a powerful heart, not a powerful here, but a powerful heart with high blood pressure. But your blood pressure can't be too high because you don't want to damage the lungs. So that results then in a double circulation pattern. One circulation circuit from the heart to the lung, and then another from the heart to the body. Each of these need to be separate so that you can have different blood pressures, so that you're not damaging the delicate structures of the lung. And you want to maintain these different blood pressures. Maintaining different blood pressures requires the formation of a septum in the heart. The early terrestrial vertebrates probably lacked this septum. We make that assumption based on the fact that some of our groups still today, the turtles and the lizards, they only have a muscular ridge. So we're going to assume that the early vertebrates, their heart had more of a spiral valve system to it, like we see in some other groups, and that is that this development of the septum actually took quite some time. Increases in blood pressure and blood flow necessitated the development of this permanent septum. Along with the permanent septum, we find that the valve systems also become different. We go from having two arches in the embryos to a single arch, but the mammals retained their left arch and the bird groups maintained their right. The single arch is better because it's going to reduce the amount of friction as blood flowing through the heart, thereby allowing blood flow to be a lot smoother and more efficient. Sorry, those words about the valves are missing from this. They're on your copy, though, of the slides. Along with all of this, we have now the ability to separate locomotion from respiration. We're able to do better respiration, bring in more oxygen. We're able to pump it around the body more. We're also doing more metabolism in order to do better locomotion. which means we're creating a lot of heat as a byproduct. So endothermy is a way to take advantage of this heat that would otherwise really be wasted energy. Increasing activity in both lineages sees some very strong resemblances. There's a change in the body form as the limbs move vertically underneath the trunk. This is linked to an increase in metabolism because of the amount of locomotion, and as I just mentioned, more food in order to do that, but more oxygen, more blood flow. Heat production is going to be a byproduct of this increased metabolism. I just want to pause here and remind you that as we learned in chapter 9, endothermy and ectothermy both work well. They're equally good, but they're going to work differently in different locations, as we'll see in chapters down the line. But endothermy is going to require heat and insulation. Through a research study done in 1958 by Roman cows, we discovered that you needed both. It isn't an easy thing, though, to get from ectothermy to endothermy, because retaining heat is going to increase body temperature, but the way to do that is not simply to add a layer of insulation. Cows put fur coats on lizards. Think about it for a moment. You have these lizards running around in little tiny fur coats. If you run around in a big fur coat, you're going to overheat most likely, unless it's winter. We know that the more layers of insulation we put on, the warmer we get. That's because we're endotherms. Lizards are ectotherms. When he put the fur coats on the lizards, their body temperatures dropped. They didn't stay warm. They actually, these coats kept them from getting warm. That's because it interfered with their ability to get heat from the environment by actually insulating them from the heat from the environment. This kind of brings up a catch-22. You need both insulation and high metabolism in order to become an endotherm. And neither works on its own. So what we see is instead a stepwise fashion. A little higher metabolism, a little bit more insulation. Well, that's beneficial. Let's put a little bit higher of metabolism and some more insulation, and so forth, so that they're building upon each other and that there's a positive feedback relationship between them that is driving this evolutionary pathway. But it's taken us a long time to even come close to understanding this, because it wasn't until late in the 20th century that we started looking at the role that locomotion might play in endothermy. Now, synapsids, well, we know that they're endotherms. And in order to be endotherms, they have to eat a lot, they have to move a lot, they do a lot of metabolism. They're also going to need to have a lot of oxygen. And respiration for both of our groups is a really easy way to lose heat, but also to lose the water that we need in order to thrive. And we're going to need to recover the water and the heat when we exhale, because the lungs would just dry out if air just moved in and out all the time. And if we don't heat the air and moisten it as we breathe it in, it's actually more difficult to extract the oxygen from it and requires more effort, more work. We lose energy then. But then as you exhale, you don't want to lose all that water and all that heat. You want to retain that back. So we're going to need a way in order to do that. As your metabolic rate increases, recycling heat is more difficult and bringing in oxygen becomes more necessary. So suddenly nasal passages that are just these open passages, these open kind of air sacs, are not going to be sufficient to warm the air, to humidify it, and then to recover the water and cool it as you exhale. So the structures that developed that are necessary for this are the turbinate structures, or the conchae. They are thin sheets of bone in these nasal passages covered by moist tissue. We have both olfactory and respiratory structures in here. The olfactory contain your olfaction sensory cells, and they are located above and behind your nasal passages. Both olfactory and respiratory structures have air passing over them with each breath, but they're going to do different things. The olfactory ones are going to take information and pass it through to the olfactory lobe of the brain. The respiratory ones are going to warm and humidify air as you breathe in, and then as you breathe out, they're going to cool and evaporate the water from the air. This allows these structures to recycle the water and heat as air flows in and out of their structures. These are very thin sheets of bone, allowing lots of surface area for air to move over. Without these structures, it would potentially be too expensive to breathe, and our ability to do lots of locomotion and lots of things will be greatly decreased. These are raccoon turbinate structures, giving you an idea of all of those bone layers that are in there. Our synapsids also needed to have enough energy to become endothermic. This required high metabolic rates, which requires high rates of food consumption and rapid digestion to support. So in order to do this, the advances that we're going to see in digestion are that the teeth become more complex and that they're going to shred the food into tiny pieces. In order to do this, the jaw muscles become larger to provide more force. Food, when it gets to the stomach, will then digest more rapidly because it's in small pieces. That surface area to volume idea. This is an advance, and if you're not sure why, think for a moment about the sharks that you dissected. When you open their stomachs, some of you found whole organisms. Those are going to digest pretty slowly in the stomach with just the digestive enzymes and everything. But if it's instead masticated food with our teeth, our jaw, that we're masticating, chewing our food, that is going to digest a lot faster because it's much smaller. We're also going to see insulation and synapsids is important in being an endotherm. We have hairy pelts for most of the synapsid groups, but then some are like us, where they're bare-skinned, with a little bit of hair and some subcutaneous fat as the insulation. And aquatic ones have a thick layer of blubber. There does not seem to be any direct evidence of insulation in early therapsids, but we'll see that's different from what we see in the sauropsid group, where they do have some evidence of early insulation. Which brings us to our endothermy in the sauropsids. They use pretty similar solutions as the synapsids for many things, but they accomplish it a little differently. So they do need to grind up their food into small pieces to dissolve it. And they do this not with teeth, because teeth were never really complex in dinosaurs. And birds don't have teeth in their beaks. So what birds do is they eat gravel. And this gravel is maintained inside of a muscular gizzard, where the gizzard is actually going to use the gravel to grind the food, along with the muscle contractions, into small pieces that are then digested in the digestive tract. We have found fossil evidence of dinosaurs who swallowed small stones and gravel, indicating that this is an ancestral trait in here to support that high metabolism. Birds have turbinate structures. Unlike ours, theirs are made of cartilage. We believe this is a way to reduce their weight for flight, but we assume that early dinosaurs and early birds probably lacked these structures. But we assume that dinosaurs likely had moist surface areas in their nasal passages anyway, even though it was not likely to be these cartilaginous turbinates. For most of our birds, feathers provide their, well, for our birds, feathers are their insulation. But theropod dinosaurs were also endothermal. And we see that feathers developed much earlier than flight did. Dinosaur fossils contain lots of evidence of feathers. And many different types of feathers are found in there. Down feathers are the ones that are used for insulation, but some of our earliest feather fossils are not down feathers. In fact, they are veined feathers. These veined feathers are not good for insulation. They're good for lift, for propulsion, for flight, for steering. But what they're really good for are social signals. Feathers did not necessarily develop as insulation and likely did not develop as insulation. Those down feathers are probably a derived feature for insulation. Feathers were modified for flight and insulation, but were first likely used in social displays. Because of that, they were also likely colorful. There have been a few feathers recovered in amber that display different colors in them that are dinosaur feathers. So that idea that these are colorful feathers used for social displays is not very surprising when we consider the fact that birds today with colorful feathers often use them for social displays. So insulation allows you to retain your metabolic heat. We mentioned the hair and the feathers are our two modes. They are both made of keratin. Alpha keratin is found in hair, claws, fingernails, hooves, horns, and alpha is also found on the hinges of scales. But most of scale surfaces and feathers, most of feathers are beta keratin. The synapsids did not, however, ever have this scale covering of their skin. We had fur. This might be today why we have soft skin, but it's also likely why the synapsid group developed glands. Skin glands are not an option when you're covered in a hard substance like scales. They are an option when you have soft skin. The absence of glands is really kind of bad for sauropsids because they can't sweat. But it also means they couldn't develop mammary glands. Mammary glands produce a milk-like substance. We know that. But there are some birds, pigeons in particular, that do produce a milk-like substance to feed their young. But they can't produce it in the same way that mammals can. They actually produce it in their digestive tracts. Mammals produce a milk-like substance from mammary glands, or a milk substance from mammary glands. But we don't believe that the original purpose of mammary glands was to produce milk, as much as it was to help to keep their eggs moist. And it resulted then in the mother staying with eggs in underground burrows. This difference in skin led to a development or a lack of a development in mammary glands, which then resulted in different social behaviors. In your synapsid group with the mammary glands, it is the mother who is generally going to be the caretaker of the offspring because they need access to her because of the mammary glands. Whereas in the sauropsid lineage, both parents are going to care for the offspring and both provide a lot of care. And the male sometimes takes on more of the care than the female does. It's kind of strange to think of this insulation difference resulting in different social behaviors and parenting behaviors. But it does. And we also find that there's a difference in fat storage too. Not really related to the same thing, but in how fat is as an insulator is stored. The sauropsids group store it in their abdomen and their tail, whereas our synapsid groups store it throughout the body, and gender differences exist in where fat is stored, which is related to the development of the mammary glands. I'm very briefly going to mention the kidney and the bladder and the idea of waste. Your textbook spends a lot of time on this. We're not going to. High rates of locomotion and activity led to high rates of metabolism and more feeding and digestion, which resulted in higher rates of nitrogen waste production. Urine is an ancestral characteristic of ammonites and probably all nathosomes, but metabolism produces ammonia as a byproduct. Well, you can't keep it around, and you can't diffuse it away into water, so our two groups had to find a way to deal with it. The synapsid line converts it easily to urea. They then accumulate urea, concentrate it into urine, utilizing the kidney to recover some water, and it's a very effective way to get rid of nitrogen waste. The sauropsids convert it to uric acid, which is insoluble. They precipitate it with sodium or potassium to form sodium or potassium urate, and they recover all of the water from this precipitate. In case you did not know, the white bird mess on your car, that's this uric acid salt, the urate salt. That is not bird poop. The other stuff in the white part is bird poop. Senses are our next to last big thing that we're going to talk about. Actually, probably our last big thing that we're going to mention. The synapsids and the sauropsids, as we go through each of the groups, we'll spend more time on senses, but it's important to know that there are different sensory organs and different sensory focuses. The synapsids, as a group, focus on scent with poor vision. Your primates are the exception here, who have good vision and poor olfaction, we believe based on our small snout. The sauropsids have really good vision and generally poor scent, but there are some exceptions. Vultures have a very good sense of smell. Our sensory systems are going to impact the social behaviors that develop in the groups, as well as the reproductive structures that develop in the groups. Scent marking will be important for synapsids. Visual signals and colors and patterns will matter more for sauropsids, and in places where colors and patterns don't work, songs will be important. For vision, it's going to be based on rods and cones, and hopefully you know that rods are sensitive to low light, but provide poor images, whereas cones are colorful, or detect colors, and require more light, but produce a sharper image. Good color vision is ancestral. It's seen in the fish. Mammals commonly see in black and white, but there are some with trichromatic vision. The true trichromatic vision that we have is only seen in humans, apes, and monkeys. And we believe that its development is likely tied to that loss of smell and that smaller snout. So this sense instead took over with more vision reliance. The sauropsids use a lot of cones. They also use oil droplets, which we'll come back to later as an idea. But oil droplets are also found in turtles, monotremes, and marsupials, along with the birds. So it's likely that they are an ancestral quality of our groups. But these oil droplets will work by filtering wavelengths out to provide better visual images. It usually gets rid of short wavelengths that are useless in the vision. Chemiosensation can be divided into gustation, taste, and olfaction, smell. Taste buds form clusters. They've come from the embryonic endoderm. You know that they are pores that are opened to the exterior, and as you eat something, those pores come into contact with the food. In mammals, they're found on the tongue and throughout the oral cavity. For sauropsids, they're found on the back of the tongue as well as on the palate. Hopefully you know that our taste buds include salt, sour, sweet, and bitter, as well as umami, which is a savory-like taste. There may be other tastes. This is an area of active research. Olfaction cells develop from the neural crest. They're distributed in the olfactory turbinates adjacent to your nasal passages, and they detect odorants. We have no idea how they distinguish so many odors. There are not enough specific receptor proteins for every odor that there is. We know that combined olfaction and gustation give us a truer taste of the food. And that's why sniffing, eating are usually well developed in the synapsid group. Hearing is developed in both the ammonites and the non-ammonites. They're developed a little bit differently. Within the tetrapods, the cochlea is our inner ear structure for hearing. And the two groups we've been talking about in this chapter have middle ears that evolve very separately. The mammals with the 3 bones of the stapes, the anvil, and the hammer, and the sauropsids only having the stapes. But there are separate pathways of development for this within that group. And our last thing that I'm mentioning deals with the brain enlargement. As you look at these groups, you can see that certain portions of the brain are enlarged differently for different groups. But as we move away from the bony fish towards the birds and mammals, we see a shrinking of that optic tectum and an increase, a significant increase in the cerebrum and the cerebellum. All ammonites have an enlarged forebrain. The telnocephalon is especially enlarged, and we find that birds and mammals have large brains in proportion to their body size, far more so than the primitive ammonites. We also have that distinct cerebrum structure. Depending upon the group you're looking at, either the birds or the mammals, they enlarge different sections of it depending upon their life cycles or their lifestyles. We find that there's a lot of wrinkling to the brains too, and that the wrinkled surface is very important in the neocortex for high levels of mammals. And higher levels of complex behavior and conscious experiences can be found in these animals with these large, wrinkly brains. But we've discovered that the size of the brain isn't the only thing that matters. What matters is not that overall size, but the size of the brain in relationship to the size of the body. Because size of brain actually scales negatively with allometry. Larger animals have proportionally smaller brains than do smaller animals. We don't really understand why, but we know that it's not just that you need a bigger brain because you're larger. Because some of the same functions are the same regardless of size. So not every part of the brain needs to get larger, but some sections do get larger with an animal. We find that what really matters is what's called the encephalation quotient, or the size of the brain relative to the body. This helps to give us that actual size compared to body mass of the brain. So you don't have the problem that we have comparing a mouse and an elephant, where the mouse brain is actually pretty big compared to the elephant brain. But when you look at their body mass, there's a big difference. We found that evolution doesn't just cause brains to enlarge. And that there's small-brained animals with really complex behaviors and really amazing abilities in learning. We find when we look evolutionarily, isn't just that we've developed larger brains. It's actually that we have a larger spectrum of brain size that we have developed. The average brain size of animals has increased evolutionarily, but this larger brain size indicates, this larger brain size range indicates that it's not simply about the size of the brain in the head, but it is, in fact, that animals with larger brains and smaller brains can coexist well, and that encephalation complex is really what matters. And that is it for this chapter. I'm going to try and stop the recording now, but thank you for watching.